Hi friends, welcome once again in another video lecture of Easy Life Science. Here I am talking about important questions, answers and explanations of set life science exam of part 3rd. So please subscribe my channel Easy Life Science, like my video, share it to your friends and press the bell icon so that you will get a notification of my newly uploaded video. So let's start. We're continuing from question number 72. The question number 72 is the lysosomes are present in all except. Lysosomes are present in muscle cells, acinar cells and hepatocytes. But the lysosomes are absent in RBC, that is a red blood corpuscle or erythrocyte. So option C is the answer of question number 72. Then question number 73. Which of the following is a function of a lysosome? Autophagy, autolysis and a digestion. These three are the functions of a lysosome. The lysosome carry out intracellular digestion. It removes dead cells. It has a role in metamorphosis. It helps in protein synthesis. The lysosome plays important role in osteogenesis. The lysosome function in autophagy. Autophagy means self-eating. It is a catabolic process in a eukaryotic cell that delivers cytoplasmic components and organelles to a lysosome for a digestion. So autophagy, digestion and autolysis, these three are the functions of a lysosome. So option D, all of the above is answer of question number 73. Then question number 74, lysosomes are involved in. Lysosomes are involved in extracellular digestion as well as intracellular digestion. So option C is the correct answer of question number 74. Moving to the question number 75, lysosomal membrane is rich in. The lysosomal membrane, it is a single layer and made up of a phospholipid bilayer. It is rich in a sphingomyelin and a sialic acid. Lysosome contains a wide variety of hydrolytic enzymes including nucleases, phospholipases, phosphatases, proteases and lipases. So the option B, that is a sialic acid, is answer of question number 75. Lysosomal membrane is rich in a sialic acid. Question number 76. Who identified a lysosome? Novikov is a scientist who discovered and identified a lysosome. The scientist Christian D. Dewey discovered a lysosome in 1955, but the lysosome were named and observed under microscope by a scientist Novikov in 1956. So option A is the answer of question number 76. The another option, that is option C, that is Pallade. In 1955, George Pallade discovered ribosomes. Option B, that is Claude. Albert Claude, he gave techniques for cell fractionation. Option A is the correct answer for question number 76. Then question number 77, all of the following has ribosomes except. Chloroplast, 70S ribosomes are there. Mitochondria, 70S ribosomes are there. Cytoplasm, in case of prokaryotic cytoplasm, 70S type of ribosomes are there. In case of eukaryotic cytoplasm, 80S type of ribosomes are there, but the nucleus is without ribosomes. So option A is a answer of question number 77. Then moving to a next question 78. In 70, yes ribosome, yes stands for a Swedenborg unit. Option C is the answer of question number 78. Ribosome is a complex molecule found in a living cell, and ribosome it is helpful for a production of proteins from a amino acid during a process of a translation. The George Palladay discovered the ribosomes in 1955. The ribosomes are associated with a endoplasmic reticulum. Ribosome consists of two major components, the small and large ribosomal subunit. Each subunit consists of one or more ribosomal RNA and many ribosomal proteins. The Swedenborg coefficient it is given as a S letter. It is a non-asymmetric unit for a sedimentation coefficient. A Swedenborg unit measures the particle's size indirectly based on its sedimentation rate under a acceleration. So here the answer of question number 78 is a C, that is a Swedenborg unit. Then question number 79, 80S ribosomes are found in. The 80S ribosomes are found in eukaryotes, including plants, animal and algae. Question number 80, the subunits of 80S ribosomes includes. Here in this image, the prokaryotic 70S ribosome is made up of a smaller unit, that is a 30S and a 50S. The 30S is further made up of a 16S ribosomal RNA and a 21 proteins, whereas a 50S is made up of a 23 S and a 5S ribosomal RNAs and 34 proteins. Togetherly, the smaller and larger subunit assembly and forms a 70S ribosome, and it is present in a prokaryotic cell. The eukaryotic 80S ribosome it is made up of two ribosomal subunits. Smaller subunit is of 40S and larger is of 60S. The 60S is made from a 28S, 5.8S and 5S ribosomal RNAs plus 45 proteins, whereas a 40S is composed of 18S ribosomal RNA and 30 proteins. Togetherly, these two units 40S and 60S assemblies and forms one unit, that is a 80S ribosome. So here in question number 79, question number 80, the subunit of 80S ribosome include. The subunit of 80S ribosome includes 60 and 40S. Option C is the answer of question number 80. Then question number 81, the subunits of 70S ribosome includes 30S and 50S. Option C is the answer of question number 81. Then question number 82, which of the following ions are required for binding of ribosomal subunits? The divalent magnesium ions are essential for several aspects of a protein synthesis, including neutralizing a charge on a ribosomal RNA and allow it to correctly fold into a main unit. Removing a Mg plus ion causes ribosomes to disassemble. So, magnesium ions are required for a binding of ribosomal subunits. Option B is the answer for question number 82. Question number 83. 70S ribosomes occurs in. The 70S type of ribosomes occur in prokaryotes, and eukaryotic chloroplast and mitochondria also shows a 70S type of ribosomes. Then question number 84. Ribosomes are made up of. The ribosomes are made up of RNA and a protein. RNA and a protein. RNA and a protein. So, option B is the correct answer for question number 84. Then question number 85. The rough endoplasmic reticulum is specially well developed in a cells actively engaged in. The rough endoplasmic reticulum is responsible for a protein synthesis, whereas a smooth endoplasmic reticulum is responsible for lipid synthesis. So here question is a rough endoplasmic reticulum. Option A, that is a protein synthesis, is the answer of question number 85. Then question number 86. The nucleus contains chromosomes, and in a nucleus chromosomes are in a compacted manner. Then moving to the next question, question number 87. Plasma membrane is. The plasma membrane is selectively permeable. It allows entry of selected ions and molecules into and outside the cell. It is double membraned phospholipid bilayer. The scientist Singer give a fluid mosaic model of the plasma membrane. The plasma membrane is selectively permeable. It means it permits the transport of only certain molecules both in and out of the cell. So option B is the correct option for question number 87. Question number 88. Most accepted structural model of plasma membrane is. The most accepted structural model of plasma membrane is a fluid mosaic model given by a scientist Singer. Question number 89. Plasma membrane is composed of. It is composed of lipoproteins. Option B is the correct answer. Question number 90. Ribosomes contains maximum amount of. The ribosome contain maximum amount of RNA. Question number 91. Which structure is present in an animal cell but absent in a plant cell? The mitochondria are absent in a plant cell. Centrioles, Golgi apparatus and endoplasmic reticulum, these are common in between plants and animal cell. But mitochondria are present in animal cell only, not in plant cell. So option C is the answer of question number 91. Question number 92. A unit membrane is about, it is 75 to 100 angstrom thick. Option C is the answer. Question number 93. The enzyme which breaks up a starch into a sugar are called as. The enzyme that breaks up a starch into a sugar, that is a glucose and a galactose, it is a amylase. Amylase is a extracellular enzyme secreted by the organism like Bacillus subtilis and Aspergillus niger. Question number 94. Apoenzyme is. The apoenzyme is the protein part of the enzyme. Option A is the correct answer. Question number 95. The enzyme which breaks up a nucleic acid into nucleotides are called as nucleases. It may be endonucleases or it may be exonucleases. The endonucleases cuts a nucleic acid at a middle or a randomly, whereas a exonuclease cuts the DNA or a RNA at a ends. The option like protease, the protease acts on a protein and forms a number of amino acids. Amylases acts on a starch and forms sugars. Hydrolysis breaks a peptide bond and release a water molecule. Question number 96. A coenzyme is often a vitamin. NAD, that is a nicotinamide dinucleotide. FAD, that is a flavin adenine dinucleotide. FMN, flavin mononucleotide. And NADP, nicotinamide dinucleotide phosphate. NAD, FAD, FMN and NADP. These are the coenzymes. Also, the actual coenzyme A, these are often vitamins. So, option D is the correct answer for question number 96. Question number 97. Enzymes are named after their substrate by adding a suffix ase. Option B is the answer of this question. Suppose the substrate is a protein, then the group of enzyme will be a proteases. The substrate for enzyme is a nucleic acid, then the name of enzyme will be nucleases. So option B is the correct answer of question number 97. Question number 98. Enzyme exists in a cell as. Enzyme exists as a colloid particle in a cell. Option C is the answer of question number 98. Question number 99. An enzyme brings about reduction in an activation energy. Option A is the answer of question number 99. Enzyme lowers the activation energy necessary for transform a reactant into a product. Enzyme uncatalyzed reaction having a high activation energy. Question number 100. Which of the following statement is not correct? So ignore this question. Question number 101. Who discovered the blood groups? The blood group system or ABO blood group system discovered by a Carl Landsteiner. Carl Landsteiner discovered a ABO blood grouping. The groups like group A, group B, group AB and group O. The blood group is determined by the genes you inherit from your parents. Each group can be either Rh positive or Rh negative. So option D is the answer. Carl Landsteiner discovered the blood grouping or ABO blood grouping. Then question number 102. Enzyme bringing about hydrolysis of esters and peptides are. Option C, that is the hydrolysis. The enzyme which brings about hydrolysis of esters and peptides are hydrolysis. 6 categories are there: oxidoreductase, transferase, hydrolase, lyase, isomerase and ligase. The oxidoreductase having a function of a transfer of electrons and they results in a change in oxidation state. For example, dehydrogenase. Second type is a transferase. This group of enzyme transfers a functional group from one molecule to another. Phosphorylase transfer a phosphate group from ATP to the reactant. Kinase, this is also a transferase. Third type is a hydrolysis. Breakdown of a covalent bond using a water. A bond in between polymers like a protein or other polymers. Example, proteases and phosphatases. Fourth type, that is a lyase. Breakdown of a covalent bond without water or oxidation. Example of it is a decarboxylase. Isomerase, rearrangement of a bond within a molecule. And because of this rearrangement of a bond, the reactant and a product are having a same molecular formula, but they have a different structural formula. Means they are isomers of each other. Example of this isomerase is a mutase. Then the ligase, a formation of a covalent bond between a two large molecule. At time of a DNA formation or a DNA replication, in a lagging strand, Okazaki fragments are formed. These Okazaki fragments are joined to each other with the help of a DNA ligase. A DNA ligase is an example of this type. So the answer for question number 102 is the hydrolysis. Question number 103, aerobic respiration is performed by. We know that the aerobic respiration occurs in a inner mitochondrial membrane, and aerobic respiration is responsible for a generation of energy in form of ATP. The electron transport chain is located in a inner mitochondrial membrane, and so ATP is formed. So option B is the correct answer for question number 103. Question number 104, bile reduces the surface tension and causes. The bile causes emulsification of fat, digestion of a fat, and absorption of a fat. All of the above is the answer. That is option D is the answer of question 104. The next question 105, dialysis causes. The dialysis causes a separation of colloids from crystalloids. During a kidney failure, the dialysis carry out which separate colloids from crystalloids. Option A is the answer of this question number 105. Then question number 106, ultrafiltration helps in. The ultrafiltration helps in a formation of glomerular filtrate. Option A is the answer of this question. The ultrafiltration is the membrane filtration technique used to remove the dissolved and a colloidal material in a transmembrane. The dissolved metal ions, as their low molecular weight or they are hydrated ions, could easily transmit a ultrafiltration membrane because their membranes have a pores that are greater than a dissolved ions. The process of ultrafiltration occurs in a nephrons of a kidney. As in this process, blood flows through a glomerulus that is surrounded by a Bowman's capsule. The hydrostatic pressure forces the small molecule to leave the glomerulus and enter in a Bowman's capsule. So here in a ultrafiltration, there is a formation of a glomerular filtrate. Option A is the answer of question number 106. Then moving to the next question, question number 107, pH of RBC is lower due to. The pH of RBC is lower due to a Gibbs-Donan effect. Gibbs-Donan effect or a Donan's effect or a Donan's law or a Donan's equilibrium. This is a phenomenon. In this phenomenon, behavior of a charged particle near a semi-permeable membrane that sometimes fail to distribute ions evenly across the two sides of a membrane. The usual cause is the presence of a different charged substance that is unable to pass through the membrane and thus create an uneven electrical charge. This is a Gibbs phenomenon. The other option, that is a sodium potassium pump. The sodium potassium pump, it is a pump or it is an enzyme found in a membranes of all animal cell. For every ATP molecule that pump uses, the 3 sodium ions are exported and 2 potassium ions are imported into a cell cytoplasm. This pump helps to maintain osmotic equilibrium and a membrane potential in a cell. The sodium and potassium moves against a concentration gradient. The pump maintains the gradient of higher concentration of sodium extracellularly and higher level of potassium intracellularly. This is about the Donan's equilibrium or a Gibbs-Donan effect. The unequal distribution of a ion in a cytoplasm and exterior of a cell. This is about the sodium-potassium pump. A sodium-potassium pump is located in a cell membrane of a eukaryotic cell. The 3 sodium ions are there in a cytoplasmic side of the pump. 3 sodium ions get exported out when 1 ATP get hydrolyzed. For this, energy is required. It is an active transport because as this movement is against a concentration gradient, ATP breakdowns and 3 molecules of sodium are exported out of the cell. Simultaneously, 2 molecules of phosphate are imported in a cell. 3 sodium get out, 2 potassium molecule get in, and 1 ATP get hydrolyzed. And this is the sodium potassium pump. The pH of RBC is lower due to the Gibbs-Donan effect. The RBC, the red blood corpuscle, alters a blood pH by catalyzing a reversible reaction of a formation of a carbonic ion from a carbon dioxide through the enzyme that is a carbonic anhydrase. And because of the production of a carbonic acid in a RBC, the pH get lowered. pH also controlled by CO2 binding to a hemoglobin instead of being converted to a carbonic acid. So option B is the answer of question number 107. Gibbs-Donan effect creates. The Gibbs-Donan effect creates the osmotic pressure. Question number 109. Transmembrane potential results due to. A transmembrane potential result due to a Donan effects. Option A is the answer of question number 109. Question number 110. Rate of diffusion of a substance depends upon. The rate of diffusion of a substance depends upon a concentration gradient of a solid. Option B is the answer of this question number 110. Question number 111. Gibbs-Donan effect is seen on the uneven distribution of diffusible ions. Option A is the answer of this question. Question number 112. Nernst equation deals with. The Nernst equation deals with amount of a potential difference. Option D is the answer of this question. Then the question number 113. Homeostasis means. The homeostasis means maintenance of relatively constant internal conditions. For example, your body shivers to maintain a relatively constant body temperature when external environment get colder. So option A, that is a control of internal environment of a body, or to maintain a equilibrium of a internal environment of the body. Option A is the answer of question number 113. Question number 140. Diffusion is more rapidly in. The diffusion is more in a gas. Option C is the answer. Then liquid and then solid. Diffusion is faster in a gaseous because the particle in a gaseous moves very rapidly, as intramolecular pace is more in a gas and it is very slow in a solids. Then question number 115. In osmosis, movement of dash dash occurs through a semi-permeable membrane. In osmosis, movement of a solvent, that is option A, occurs through a semi-permeable membrane. Then question number 170. Osmotic pressure across the capillary wall is exerted by. It is exerted by a concentration of a molecule. Option C is the answer of this question. Question number 118. Effect of temperature on viscosity involves. Effect of temperature on viscosity involves. The temperature can increase the viscosity or it can decrease the viscosity. So option D, both A and B is the correct answer for question number 118. The ultrafiltration takes place in the presence of. The ultrafiltration takes place in the presence of the hydrostatic pressure. It occurs in a glomerulus. Question number 120. pH means it is a negative logarithm of H plus ions. Option B is the correct answer. Question number 121. Microcytic anemia develop in. The microcytic anemia happens when the RBC are a smaller size than a usual because they don't have enough hemoglobin, and we know that the hemoglobin is an important protein which helps to carry the oxygen throughout the body, and it is because of the microcytic anemia is because of deficiency of vitamin B12 and folic acid. Option C, both A and B is the correct answer of question number 121. Then question number 122. Extrinsic system of a blood clotting is initiated by. Option A, that is a factor 3. These factors are represented by a Roman letters. The factor 3 is a first factor which initiates a extrinsic system of a blood clotting. The coagulation cascade or a secondary hemostasis is a series of steps in response to a bleeding caused by a tissue injury. Coagulation consists of a three pathway: extrinsic, intrinsic and a common pathway. The extrinsic pathway involves initiation by factor 3, that is a tissue factor, and its interaction with a factor 7. Whereas the intrinsic pathway, the factor utilized are factor 12, 11, 9 and 7. A common pathway utilizes a factor 10, 5, 4, 1 and 12. So answer of question number 122 is option A, factor 3. Question number 123. One of the following is not anticoagulant. It is a complicated question, so skip it. Skip the question. 124. Following are membrane bound cell organelles. The membrane bound cell organelles are endoplasmic reticulum, lysosome and perioxosome. Ribosomes are not membrane bound organelles. Option C is the answer of this question. Then the question number 125. The intrinsic factor present in a cell membrane mainly functions as. The intrinsic protein, not factor, intrinsic protein present in a cell membrane mainly function as an enzyme. For example, the sodium potassium pump or sodium potassium ATPase pump, it is an enzyme. So option A is the answer of this question. So thank you for watching me. Subscribe to my channel Easy Life Science, like my video, share the video to your friends and press the bell icon so you will get the notification of my newly uploaded video. I am going to upload the part 4 of this series. So press the bell icon that you get the notification of that uploaded video. Thank you once